Hey, how are you doing?
Hey, how are you?
I'm good. Let's wait a few more minutes before getting started.
Yeah, no problem. Also, do you have like a group me by any chance?
No, what?
Do you have like a group me for
A
MCAT
group?
Max?
I don't think so.
I think it would be a good idea to have one, like so you can send reminders there and people can,
Oh yeah,
because
true.
everyone
Uh,
would share it with friends, I guess.
Yeah, that is good. Yeah, it's a good idea. Definitely try to start one. What's up, Samir? How you doing?
How you doing, man?
I'm doing good.
It's good to see you.
Yeah, it's good to see you as well. So your last semester at VCU, right?
Yeah. When are you graduating?
I'm graduating this semester as well. So,
Okay, nice.
yeah.
Cool. When's your graduation date?
I don't even know when I'm graduating to. Like, is there an official date for graduation?
Yeah.
Oh, I don't
Do you
know.
do you have your cap and gown and stuff?
I didn't even order a cap and gown or anything like that.
Go to Barnes and Nobles. They have them there. You could pick them up right now.
Oh, really nice.
Yeah, it's based on your height, so it's like customized.
Oh, nice.
But yeah, my walk day is the 12th, the same day I'm taking
Well,
my MCAT.
oh, really?
Yeah,
So,
I'm gonna take my MCAT in the morning after walk, and then I'm done basically.
dang.
Yeah.
Wow.
So I'll really be finished. You know what I mean?
That's gonna be a really big day. Wow.
I know, right? Right.
Yeah. Dang. All right. So I guess we can go ahead and get started. Let's see. Okay. Can everyone see the screen?
Yep.
All right. So today we're going to be walking through like a passage from the biology section from the MCAT. And so this passage is called GnRH and Kisspeptins. All right. I guess we can kind of skip the icebreaker today. It's not that many people to, I think everyone like kind of knows each other. Or like, okay, so kind of like the main topic of today is this thing called the hypothalamic pituitary gonadal axis. And this is kind of like the pathway that's responsible for releasing most of the sex hormones in your body. So for like males, this is responsible for releasing testosterone. For females, like estradiol, progesterone. So kind of how the pathway works is you have neurons in your hypothalamus in your brain, and they release hormones. So if we're just going to look at reproductive hormones, so how it all starts is from your hypothalamus, you have this thing called gonadotropin releasing hormone, and it goes and acts on the anterior pituitary, and it causes the anterior pituitary to release either LH or FSH. And so these are called gonadotropins. So that's why it's called gonadotropin releasing hormone, because when the gonadotropin releasing hormone goes to the anterior pituitary, it causes gonadotropins to be released. So that's why it's called GnRH. So LH and FSH are called gonadotropins. And so these ones go and act on your gonads, so like testes or ovaries, and they cause testosterone or estradiol / progesterone to be released. So that is the main like hypothalamic pituitary gonadal axis. So does anyone have any questions about that? Yeah. All right. Okay. So now we're gonna do some like analysis of a table that's gonna be in our passage because it's a tad bit complicated, but if we break it down, it becomes a lot easier. So let's say we have like this compound, we're going to call it kisspeptin, and we're going to say it can alter this pathway, right? So I guess the first question where we want to start is how does kisspeptin affect the GnRH and LH levels? Just based on the table. So
Seems
saline
like
is just we are control. And then how does the kisspeptin affect GnRH and LH?
seems like it increases GnRH, which subsequently increases LH.
Right. Yeah. So that's actually begs the question. So we know for a fact kisspeptin increases GnRH, and we know that GnRH goes and acts on the anterior pituitary to release LH, right? So we want to figure out, is the increase in LH here just because of the GnRH increase, or is it because kisspeptin is going directly to the anterior pituitary and acting on it to release LH? So we don't really know which one it is. We don't know if it's just because of GnRH or is it directly acting. So is it acting up here to increase GnRH or is it acting down here? We don't know which one it is. So we need more information. So thankfully they gave us another table here. So what we did right now is we created some mutated mice or rats. And these rats can't release GnRH. They're just like mutated, so they can't. So now we've taken away kisspeptin's ability to increase GnRH levels. So we can figure out, is it really directly acting to increase LH levels, or is it just relying on increase in GnRH to increase LH levels? So we can kind of distinguish now. So what we can kind of do is we can look at this first row right here called GnRH alone. And this gives us like a baseline level of GnRH and LH, right? And so that's how our GnRH and LH levels should look, right? So now we're going to try adding kisspeptin and GnRH, the middle row. Do our GnRH and LH levels change?
Not really.
Yeah, there's really no significant difference. So we've taken away. So remember, these mice can't release GnRH. So we took away the mice's ability to release GnRH.
I have a question. If it's true that the mice can't produce GnRH, then the GnRH we're adding, like for example, in the GnRH alone one, we're adding 0.50.
Yeah, exactly.
Okay, I see. Okay.
Yeah. So we're just supplying them with the GnRH. So Kisspeptin can't affect the GnRH levels. So we've taken away that ability of it. So now that we've taken away that pathway for it to increase GnRH levels, the only way by which it could potentially increase LH levels was by directly acting on the anterior pituitary gland to release LH. So that's the only way it could increase the LH levels. And when we take away its ability to increase GnRH, there's no increase in LH levels. So what does that tell you?
It doesn't act on either.
Yeah, so it does act on GnRH usually, but we've taken away that ability. But
okay, I see.
But now it can't we know for a fact it doesn't directly act to increase LH. Because when we took away its ability to increase GnRH, there was no increase in LH levels. Remember, we were trying to figure out, does it just increase LH levels because it releases GnRH, or does it go directly act down here?
So in other words, it only doesn't affect either in this case because we can't test if it's affecting GnRH because the mouse can't make it.
Yeah, so
we've confirmed that it can't doesn't act on LH.
Right, we confirmed that, and the whole reason we took away the first option is because we wanted to. We knew it increased GnRH normally, but we didn't know if it also did the LH. So we wanted to take away the first option, so to see if it still increased the LH levels when that first option wasn't available. So that's kind of what they tried to do there. Okay, so now we have let's say we have another compound, neuropeptin Y, also MPY. So when you add MPY and GnRH, what happens to the levels of each hormone?
Has no effect when it's
LH increases.
Yeah, LH increases, right? So remember, we've set up the mice so that it's impossible to change the levels of GnRH, right? So there's no way they can increase LH levels just by increasing GnRH. So how can it increase the LH levels then? What's the only other option?
Adding NPY.
So like how does MPY change? So remember, we have two ways. They have two ways they can increase it. You can either increase it by increasing GnRH, or you can go directly and act to release LH.
Outside directly.
Yeah, directly. Because remember, we've taken away the ability to increase GnRH here.
Mm-hmm.
So the only way you can increase the LH levels is going and directly acting down here, the anterior pituitary, to increase LH. So based on that, what's the difference between Kisspeptin and MPY?
Kisspeptin only affects GnRH and then NPY affects LH.
Right. So Kisspeptin does not directly affect LH. It affects GnRH, which in turn affects LH. It's a bit more indirect, right?
I have a question. Why doesn't the NPY alone affect the LH?
So because you still need some GnRH
to like get it started.
Yeah, exactly.
Okay. All right.
Yeah. So you still need a GnRH, but we know that MPY directly acts to help release LH, right? So that's the difference between Kisspeptin and MPY. Kisspeptin, we know Kisspeptin, when it can't increase, when you strip away its ability to increase GnRH, it's unable to affect the LH levels. So we know the only way you can actually increase LH levels is to increase GnRH. And the neuropeptin Y, even when you strip away the ability to increase GnRH levels, we still have higher LH levels. So that must mean it can go and directly act to release LH. Okay. So that's kind of breaking down the table. Yeah. And so we talked about MPY. Okay. Okay, now this is another thing we need to talk about. So I think, we transcribe DNA, we make pre-mRNA, And then we splice the pre-mRNA to make mRNA, right? So so the important thing to note is just because something like, for example, exons, they get to stay in the mRNA, the introns get spliced out. But just because something gets to stay in the mRNA, like an exon, doesn't necessarily mean it's actually going to be translated. So there's actually regions of mRNA called untranslated regions. So 5' UTR, 3' UTR. So for example, if the passage says there's a protein whose exon is in the mRNA, yet it's never translated, that would mean that DNA for the exon is in the UTR region, right? So if an exon's still in mRNA but never gets translated, that means it's in the untranslated region. Okay. And then so 8. So this is for the MCAT, you have to know a bit about the menstrual cycle, but not too much in depth. So the biggest thing we want to focus on is that we want to kind of separate it into two halves. So pre-ovulation and post-ovulation. So ovulation is when an egg kind of like bursts from the follicle. And so what happens before that for the MCAT, what you need to know is that you have a peak estrogen rise and peak luteinizing hormone, LH, rise before ovulation. And so those peak right before ovulation, and the bursted follicle now becomes this thing called the corpus luteum, and that thing releases progesterone. So you want to know pre-ovulation, you have an estrogen LH peak. Post-ovulation, you have a progesterone peak. And so progesterone kind of like thickens the uterine wall. So blastocysts, which is like a fertilized egg, can implant. So that's what progesterone is for. And estrogen and LH are involved with like ovulation. So you just want to know pre-ovulation, which is also called the follicular phase, you have rises in estrogen and LH. In the luteal phase, you have progesterone peak. So just got to remember that. Okay, so we know that. And then this is kind of basic. So, to break proteins are held together by like peptide bonds. And so if you want to break a protein apart, you got to add hydrolase. So you add a water. The enzyme uses a water molecule to break apart the bonds, right? So does anyone have any questions about that background PowerPoint? You good? All right, sounds good. So let's move on to the passage. Okay. So everyone can see my screen, right? Okay. So does someone want to read this first paragraph? It's kind of short passage.
I can read it.
Yeah.
Gonadotropin hormone releasing hormone, GnRH, is the key regulator of the reproductive axis. GnRH is a decapeptide, glue, his, trip, serine, tyrosine, glycine, leucine, arginine, proline, glycine, NH2, released by the hypothalamus and which is responsible for the secretion of gonadotropins, a class of hormones which are directly responsible for sexual maturation. During development, the defective migration of GnRH neurons into the hypothalamus causes Kalman syndrome, characterized by hypogonadotropic hypogonadism, defective gonads, and anosmia, loss of smell. Researchers investigating.
So Devian, do you want to read the next half?
If you can, I'm on the third floor. I wouldn't want to.
Oh yeah, no problem.
Loudly.
You want to read it, or do you just want me to read it?
Researchers investigating potential treatments for Kalman syndrome looked at how NPY and Kisspeptin impact the reproductive axes. NPY and Kisspeptin are both known modulators of GnRH signaling. The Kisspeptin gene, has 4 exons, the first 2 of which are never translated. The gene encodes the precursor 145 amino acid peptide, which is cleaved to a 54 amino acid protein. A study examined the effect of Kisspeptin and NPY on the levels of blood GnRH and LH in wild-type female rats with a mutation preventing the secretion of GnRH.
Okay. So you have these tables here. So let's try question one. Question one says, which conclusion is best supported by the data in the passage? So number one says kisspeptin directly amplifies GnRH secretion. Number two says kisspeptin directly amplifies LH secretion. And number three says MPY directly amplifies LH secretion. So what is the correct answer to this question?
C.
Exactly, right. Because again, we know for a fact kisspeptin increases GnRH levels. We didn't know whether if the increase in LH levels were because of that GnRH increase, or if kisspeptin actually directly amplified LH secretion. So we didn't know that, but then we look at table 2. We see we have a baseline level of GnRH and LH, and then we add kisspeptin to that, and nothing changes. So that must mean that kisspeptin doesn't directly amplify the LH levels, because if they could, then those LH levels would change, even though the release of GnRH was not changing. So we knew that. Then we looked at MPY, and MPY was able to directly amplify LH secretion, because when we took away its ability to increase GnRH, it was still able to increase LH. So that's how we figured that out. Just a recap. So yep, and that's correct. Okay, question 2. Which of the following is the most likely the reason that the first 2 exons of KISS1 are never translated? And so let's actually look back at the passage and see where it talks about the KISS1. So it says right here, the KISS1 gene has 4 exons. The first 2 which are never translated. Okay. So if these are the first 2 exons, and which way is mRNA translated?
Would it be C, 5' UTR?
Yeah, exactly. Because we always go 5' to 3' for translation. So first 2 is going to be towards the 5' end. That's correct.
Why is it not D? Isn't D also true? Because you said
so
they don't.
They always do code for amino acids. Like if there's mRNA, there's always going to be tRNA that can bind and bring in amino acid.
Okay.
So they do technically code for amino acids, but they just like never actually get translated.
Okay, so they code for amino acids, but they don't get translated. Can they get translated?
If they're in the untranslated region, no, they can never get translated.
If they can never be translated, how can they code for amino acids?
I mean, they code for it, but like they never actually
Is that like a valid question or like?
I guess just a bit like weirdly written question. Yeah.
Yeah.
Yeah. But
I get it.
They said was the correct answer.
Yeah.
Yeah.
We can check why D is incorrect in the solutions.
Let's see.
Oh yeah.
So stop codons are the only codons that do not code for an amino acid.
Oh.
It's very unlikely if these exons simply contain repeats of the same stop codons. Yeah. So I guess they still technically can code for.
So they can be translated, but they just usually don't, or
yeah, I guess we can think about it that way.
like maybe if there were like a mutation or something that caused like a frame.
Yeah, to move. Yeah.
Okay.
Yeah. Okay. So it says, which of the following graphs shows how the blood estradiol levels of a wild type mouse would change in response to kisspeptin injection? So if we had kisspeptin, would you get more estradiol or less estradiol?
Less? No.
So kisspeptin increases GnRH, right?
Yeah. And that increases
LH, and LH increases estradiol, right? So what would you want the graph to kind of look like?
Probably C.
Yeah. So C is the only graph that actually shows an increase. Makes sense. Okay. Elevated levels of which hormone specifically indicate that a female is in the luteal phase of the uterine cycle? So remember,
A.
which hormone did we say that is peaked in the luteal
LH.
phase?
A, right? No.
Which one?
Yes. Progesterone.
Yeah, it's progesterone.
Yes.
So LH peaks at the end of the follicular phase, so like ovulation. And then progesterone's after ovulation. So that's why progesterone. Okay. Last question. The enzyme responsible for producing the 54 amino acid protein fragment. So remember, we said that KISS1 gene encodes for a precursor 145 amino acid peptide. It's cleaved to a 54 amino acid protein. So if we're cleaving a protein, what enzyme are we going to use?
Hydrolase.
Exactly. And that is that. Yeah. It's a pretty short passage, honestly. Pretty short. Let me see. Yeah, that's it. Do you have any questions? Good.
Yeah, I have a question. I always thought that LH makes progesterone and FSH makes estrogen.
So FSH increases the growth of the follicle cells and they release estrogen. So they don't actually like make estrogen, but they kind of contribute to the increase in estrogen.
So go back to question 4 or,
Yeah.
or 3, right? I think
Yeah,
three.
that was 3.
Why would is estradiol not estrogen?
It is estrogen.
So why would estradiol increase if we're increasing LH? If LH is
Well, we're increasing Kisspep. So LH is also oh no, Kisspeptin increases GnRH, which also increases FSH and LH. Yeah.
Oh, okay. All right,
Yeah.
that makes sense.
Yeah.
Okay.
Yeah. So that's that. Yeah. Any other questions?
Wait, what was the mechanism you said for how FSH increases estrogen? You said that it goes to
So FSH is called follicle stimulating hormone. So it increases the growth of the follicles that have the eggs inside. And when it increases the growth of those cells, they release estrogen.
Okay.
Yeah.
Then in men, it's Sertoli cells, right, to make sperm.
Exactly. Yeah.
And then for FH in men, that's Leydig cells for testosterone, right?
So LH is the Leydig cells for testosterone.
LH.
FSH is the Sertoli cells.
Yes. Okay.
Yeah.
Okay.
Exactly.
Cool.
Yeah, no problem. Any other questions? All right. Well, thanks for coming out, guys. And good luck with studying.
Thank you, Sid.
Yeah, no problem. All right. Have a good day.
So did you ever what are your thoughts on Jack Weston cars?
Jack Westin cars. I've never actually done it. Isn't it pretty? I've heard people do it though, right?
I heard it's like really hard. Like I just did a passage yesterday and like I did a lot better than I did my first time, but like
Yeah.
definitely think it's harder. I'm just wondering if you had like any
So
experience.
my philosophy for cars was always to do the AMC cars
Yeah.
because no matter how closely they can replicate it, like cars is kind of like slight nuances. So
Yeah.
like my logic was like I only wanted to be trained in the AMC way of thinking because
I had that idea too, but I kind of just
yeah.
wanted to get some practice like just reading in general, you know what I mean?
Oh yeah. Yeah,
Something
reading in general is always good. But I think the AMC they have like the cars Q packs.
Yeah, I
Those are
have
pretty
those.
helpful. And also like the FL, like the full length cars, like AMC full length cars, those are also good.
Yeah, sure.
Yeah.
What's like the strategy for like let's say like you take a full length and you miss a
Yeah.
question just because like you misread the passage or something, right? Just
Uh-huh.
like it's like a stupid mistake, you know what I mean? How do you like learn from that?
So you want to like you want to kind of think back to like your thinking during that test. Like what were you thinking when you incorrectly answered the question?
Uh-huh.
And then you want to quiz yourself on like what's the correct form of thinking. So you want to like quiz yourself kind of like what you should have done. So you don't make that mistake over again. And even if it's like a stupid mistake or like just a careless mistake, you can also like write it down and like review it later. So like you can just like flip through your mistakes.
True.
Yeah. So those are some good things. But I say the biggest thing is like just trying to quiz yourself try to like
Yeah.
try to use your brain as actively as possible, like force yourself to like kind of recall stuff.
Yeah, yeah. Because I feel like it's easy to like make like an Anki card or something out of like a
Yeah.
question I missed that like is like conceptual, but like if it's like I just like literally I couldn't do 76 divided by 800 or something. Like
Oh
I,
yeah.
you know what I mean? Like it's just like
Yeah.
I'm nothing in the moment.
But even then, I feel like it's still good to review it just so
Definitely,
you can
yeah.
you still have a chance of making that careless mistake again. So
I
if you
feel
review
that.
it, then reduce it next time. Yeah.
Yeah, yeah, yeah.
Yeah.
Do you do you think it would be smart to like just have like a folder of like screenshots of every single missed question that like I've ever missed?
You could do that. It's just I don't know about because you already have an electronic version of them, right? Like you
Yeah,
have
but
the
like they're different. Like some are in New World, some are in like
oh,
one,
I
some are
mean,
in like
if
my,
you
you
have
know what I
time
mean?
for it, you could. I just like I found it kind of like time consuming to take screenshots of everything and stuff like that.
Yeah.
But what I would do is just like I would just go to it, like maybe cover it up with your hand, like try to quiz yourself, like what was the mistake I made here? Like what
Mhm.
was the question testing? Like how am I supposed to actually solve this question? Like pretend like you're doing it again. Like how am I supposed to solve it on test day?
Yeah, I find that I can see that, but I find that very hard to do for cars. What do you suggest about that?
So cars actually is very helpful. What I did is like I would always think about like how I was thinking and how
Uh,
that was wrong and how I'm supposed to think. Like I
okay.
would always just try to program myself, like quiz myself. Here's how I'm supposed to interpret the passage. Here's how I'm supposed to apply that to this question. Like I should think in this way.
And
So I
were
just
you able to like apply that reasoning to like other passages and
yeah,
stuff?
exactly, yeah. So all of it was just like kind of forcing your brain to think like the test makers because there's a lot of different ways to interpret all these questions, but there's only one correct way according to the test writers. So I always try to quiz myself on the correct way to answer the question. So I was
Mhm.
like cover up the question, be like, how am I supposed to answer this? Like what line of logic am I supposed to use to answer this question? So I answer it how the test makers want me to answer it.
I see.
Yeah, and cars that's really helpful. Honestly, I'd say that's the biggest because cars can feel like really frustrating at times because there's nothing you can really learn for cars. It's
Yeah.
just like yeah. So that was like the biggest. I feel like cars at least, and even the other sections.
Yeah. What's like a tip you learned like while reviewing cars that like that you remember like from your mistakes or something like just like a way you should have been reading something but you weren't.
I think the biggest one is like if the answer doesn't pop out as correct to you, like it doesn't immediately pop out as correct. Like if you have to force yourself to kind of like convince yourself to select the answer, it's probably not right.
Hmm.
So you want to do answers that are like most clearly supported by the passage. So answers that kind of like pop out of the page is correct. Like
Hmm.
there should be a strong evidence supporting that answer. It shouldn't just be like, I could see this being correct if I like kind of like
Yeah, yeah, yeah.
think about it a lot. It should like kind of pop out.
What was your score breakdown by the way from
I scored 132 on Chem Phys, and then 130 on all the other sections.
wow.
Yeah.
Nice.
Thanks. Yeah.
So all right.
Yeah.
Thank you, Sid.
Yeah. No
See
problem.
you later, man.
All right. Thanks for coming.
For sure. I appreciate you.
Yeah, no problem.
See you
All
later.
right. Have a good day.
You too.